an MBBS student at AIIMS Bhubaneswar. अभी मैं फोर्थ सेमेस्टर में हूँ and आज I'm gonna answer your queries related to biology. So I have already received some questions from your side, and we'll start with the first question without wasting much time. The first question is, dicots have a taproot system where the primary root grows deep into the soil, while monocots have a fibrous root system where the roots remain close to the surface. So I think कि आप इसको explain करना मेरे को explain करने के लिए बोल रहे हैं, तो start करते हैं. तो जो dicots है वो evolutionarily पहले आए हैं monocots के, so they are older than monocots and they are less developed than monocots. What the strategy in a dicot root is कि if this is the soil and root को मैं red से बना रहा हूँ, we'll start with the seed. Suppose this is the seed and you have buried the seed in soil. Soil में आपकी seed है. So your radical will grow out of the seed, go down because it is negatively geotropic. Okay? Negatively geotropic का मतलब वो gravity के opposite grow करता है. कुछ देर बाद जब radical sufficient water और nutrient absorb करने लगे, your plumule comes out of the soil, which is positively geotropic, और उस पे आपका leaves वगैरह चलेंगे. आफ्टर सम टाइम आप देखेंगे कि जो रूट है उसको आप इस तरह से ड्रॉ कर सकते हैं. एक मेन रूट है जिसको हम टैप रूट कहते हैं, और उसके उससे बहुत सारे फाइबर्स अटैच्ड है. सो द मेन पिक्चर इज देयर इज अ मेन रूट which is known as a tap root which grows pretty deep into the soil, right? And from that tap root a lot of other fibers are originating which are also assisting in absorption of water and minerals. This is a strategy which helps the dicots to grow a bit tall. Okay, if the root is deep into the soil and there are primary, then from primary you will have secondary, from secondary you will have tertiary roots. This will give good anchorage to the dicot plant, and the plant will be able to grow tall, right? If you have a good support system, you'll be able to grow tall. This is the dicot root system. We'll move on to the monocots. Now, monocots are evolutionarily more advanced and younger than the dicots. The thing is, monocots, they realize that we don't need a lot of height to, survive or to make food in a much efficient manner. What we need is, we need to be more efficient in trapping the sun's energy. So the main strategy in a monocot is we need to trap more of sun's energy. So they have what is called an they have a fibrous root system where there is no main root. There are lots of fibers originating from the base of the stem. The stem itself is not very huge or not very lengthy, but might be reduced to just a disc, as you see in case of a lot of monocot plants. The leaves are big usually and they absorb a lot of sunlight. They are very efficient in converting water and sunlight into carbohydrate as compared to their dicot counterparts. So this is a fibrous root system. Let us compare both of them. Some features which I'm going to compare are stability. So in terms of stability, I would say a taproot system is pretty much stable. I'll give it a 3 stars. In terms of stability, a monocot root system or a fibrous root system is on a 2 star. It's not That's stable, but it's not bad. It's not key the plant will easily die, but you can actually uproot the plant with much ease. So if you want to uproot a monocot as compared to a dicot, let's compare ki you want to uproot a corn plant or a wheat plant in a farm. Compare that to uprooting a mango tree or a tree in the garden. You know that uprooting a mango tree or a dicot plant will take you much more effort than a monocot plant. That's why I'm comparing the stability. Next thing, efficiency. Okay, here I would say that a monocot root system is better, because imagine that in a dicot taproot system, suppose you find water over here. Now you have to transport that water from here to a secondary roots, to the primary rootlets, to the taproot, then it goes to the stem. But in case of fibrous root system, you find it here directly. You'll be able to give it to the stem in a short path. That is called efficiency of trapping nutrients and water. Okay, it is much better in a fibrous root system, right? Then expansion or asexual reproduction, I think I'll give a 1 star to the taproot system here. Whereas in terms of expansion, a monocot root system or a fibrous root system does much better. So what I'm trying to compare here is, suppose this fibrous root goes over here, it encounters the surface of the soil, and it can grow a new plant over here. Got it? That is how they spread. It's called a runner. Okay, this thing is called a runner. There are other examples which you will learn in morphology. Okay. Whereas if you compare a taproot system, it is almost impossible for a tertiary, secondary, primary rootlet to go and establish a fucking taproot system. Okay. A taproot system is very difficult to establish by a rootlet. This is not possible. So expansion capacity is limited. Right? Okay. I think we have done enough comparisons. We'll move on to the next question, guys. The next question says, what do you mean by convergent evolution? Okay. Now, talking about evolution, it can either be divergent or convergent. Usually, when we talk about Darwin's evolutionary theory, we talk about a divergent evolution. Most of us know that we have a mental picture of evolution as a divergent evolution. What a divergent evolution is, there is a common ancestor, and it gave rise to a lot of subspecies, species, genuses. Okay, that is divergent. Now, this is normal. It is pretty easy to understand that over the time, the common ancestor must have gathered enough variations, gathered enough traits in one of its offsprings for it to be considered a new species. Then that offspring must have gathered enough traits and enough new features in one of its offsprings for it to be considered as one more new species. Now, that's how divergent evolution works. Not much to understand over here, but the question asks me about convergent evolution. So we'll take a look at convergent evolution. Now, evolution is, it's not always creation of a new species. Evolution is it's a process, it's a story which tells us how the current scenario came to be, right? How the current scenario came to be, and what are the factors which influenced the species diversity in present to be like this. For example, why, okay, evolution can answer a lot of questions. It's not just how a common ancestor gave rise to these species or how these species came to be. It's like why in deserts we have that kind of animals or plants, why in a tropical rainforest we have different kind of animal and plants than in a desert, how and what factors have influenced the desert flora and fauna to be different from the rainforest flora and fauna? All of these questions are answered by evolutionary theories. Okay. Coming back to convergent evolution, I'll give you an example. Let's imagine, before giving you the example, I will tell you what convergent evolution is in a short jinx. So always think of convergent evolution as evolution has having a goal. Suppose you want to achieve something and you evolve yourself in a way that you are able to achieve that. That is how convergent evolution works. For example, you want to fly. So there is this thing called flight. Why would you want to fly? Better reach to your food, better view. You can view dangers and threats from a long distance. Better escape. All of these will tempt you to be able to fly. Now, different species at different points of your, what do you call it, the animal kingdom. The animal kingdom has like lots of phylums, genuses and all. Suppose this is phylum 1. phylum 1 का एक genus 1 है. Suppose this is phylum 3 and इसमें एक species number 2 है. Okay. Now both of them have been exposed to such an environment where flight is going to be a game changer for their species. Okay. Flight is going to be a game changer. Now, some of them will start accumulating characteristics favorable for flight. Suddenly, they will evolve into a new species, species X. This will evolve into a new species, species Y. Now, species X, what it did is, if this is your genus 1 and genus 1 ka animal looks like this, it has 6 limbs, but species 2, it looks somewhat like a bit more elongated and it has no limbs. Let us consider that it has no limbs at all. Now, species X somehow modifies one of its limbs into a wing. Okay. One of its limbs is now a wing. The other two are the same. Species Y grows wings out of one of its appendages. For example, it has ears, right? It grows its ears into wings. Now, both of them are having something which they can use to fly. Let's take one more example. A more advanced species, phylum 5, there was a species number 1 and it evolved into species Z. This species Z, what it did is its hands, its feet, it turned them into webs and now it has got a good wingspan. Okay. Now, all three species have one thing in common. They have common attributes. They can fly. All three of them can now fly, but they do not have common ancestors. And they do not have common origins. Origins of what? Both their own and their modifications. Now, species X modified one of its limbs. Okay, it modified one of its limbs. Species Y modified its ears. Species Z has modified its appendage, like its fingers, toes. All of them have different origins. All of them have different ancestors. It looks somewhat like this. This is 1, 2, 3. All of three of them have kind of. No, initially all three of them couldn't fly. Initially, all three of them couldn't fly. Now, all three of them happen to fly. but they have different ancestors, different origins of their own, and different origins of their modifications. This is what convergent evolution looks like. Can you see the three ancestors, ancestor 1, ancestor 2, and ancestor 3, converging into one common attribute, which is flight? That is how convergent evolution works. You set a goal, you want to fly. Now, in order to fly, you modify your body in a modifiable way, and you start flying. That is called convergent evolution. That is how it works. This is the jinx of convergent evolution. Okay, guys? Okay, let's move on to the third question. Third question says, is there any new fossil discoveries or controversies about evolution? Of course, there are a lot of controversies on evolution. Okay, I'll give you one example to go with. So there's this organism called coelacanth or lobe fin. Now, this organism was considered to be extinct because it existed so long back that scientists thought that this organism cannot be like surviving in the present scenario. But they were wrong. It was found in this South Africa, and they discovered coelacanth living in South Africa. They discovered a living specimen over there. What does that mean? That means that the present picture that we have about evolution, it is always changing. Who knows, some days we might find dinosaurs or their remnants living all the way back from that era, and they have survived that era, and they're still there on this earth. Then the whole evolutionary story changes, right? This is one example which I wanted to give. Okay, moving on, functioning of the epidermal tissue. So now we move on to, away from evolution, we move on to the epidermis. Now, what is an epidermis? In general, any epidermis is the epidermis is the outermost layer. Outermost layer is known as epidermis. So it can be the outermost layer of your skin. It's still called epidermis. It can be the outermost layer of a plant, a bark. It's still called epidermis. It's a common term in biology used to denote the outermost layer of something. Suppose you discover a new entity and you name the outermost layer as epidermis, totally fine. It's gonna work. Functioning of the epidermal tissue, I consider this question to be from botany. Okay, because the NCERT describes epidermal tissue in botany much more vividly. So I will consider this to be botany and answer your question according to botany. Now, I will draw an epidermis. These are the cells of the epidermis. Usually and mostly, epidermis is a single layer of cells. And you know that a collection of cells performing similar function from the same origin are known as tissue. So now, what is an epidermal tissue? This is an epidermal tissue. How does it function? What are its modifications? That is an additional question that should have been asked, but I'm going to put it there. Now I'm going to erase some of the cells and draw some of the modifications. Now I erase this cell and I draw something called as a guard cell. You know what a guard cell guards, right? Yeah, you're right. It guards the stomatal aperture or stomatal pore. We're going to draw hairs. Okay. They're usually called trichomes. What do they do? We'll get to that. The primary function of an epidermis is, as you might have guessed, protection or making a barrier, creating a barrier between the outside and the inside environment. Why do you need that? Of course, you need to protect the inside environment from the outside environment. Otherwise, what's the difference between the living inside and the non-living environment outside? You need to do certain metabolisms, certain processes inside your cell, inside your tissue, inside the organism, which is not occurring outside in the environment. That is why you need to separate the environment from your body first. That's what epidermis does in every organism. That is the primary function of epidermis. Got it? Now, how does it do that? Of course, these cells of the epidermis, they are specialized in doing that because they stick to each other. Okay. Okay. These cells have good intercellular connections. They are tightly sticking with each other, which helps them to form a good barrier against the outside environment. So a lot of substances cannot cross them. But you want some substances to cross the barrier, right? Otherwise, how will you derive raw material for maintaining your inside environment? For that, we have these openings on the epidermis called, yes, you are right, stomata. These openings are called stomata. The cells surrounding these openings are called guard cells, which are modified epidermal cells. Now, what does stomata do? Again, you're right. Stomata helps in exchange of material between the inside of the plant and the outside. Mostly it is air and water because that's what plants can exchange conveniently. Other things which they cannot exchange with the environment, they just store it in places. And some of those places are beneficial to us, and we cut out those places, we obtain those storage sites, and we use those stored materials, right? Yeah, fruits are examples of that. Other examples are, resins and all those gums and all. Okay. Now, how does the stomata function? The guard cells, when there is less water, the guard cells become less, turgid, and they close the pore. No need for exchange of water at that time. You don't need to lose water. But when there is like a lot of water in the plant and you need to lose all of that water, the extra water, you will have to open up your stomatal pores. What happens is the guard cells become turgid, and when they are turgid, they bend. They bend and create this pore called stomata, from where exchange of water or diffusion of water can take place. That's how they work. Now, coming to these hairs called trichomes, what do they do? Simply, they prevent a lot of loss of water. Okay, if trichomes are not there, your stem will start losing unnecessary extra water. Okay. Okay, so that's it for the question number 4. We'll move to question number 5 quickly. Question number 5 asks, what is Big Bang theory? Explain Big Bang theory. Now, all of us science students, we know what Big Bang theory is. We've been like hearing about it for since we have started reading about space, any encyclopedia, all of those stuff, they mention the Big Bang theory. What is the Big Bang theory? It is theoretically considered to be the beginning of our universe. That's how everything started. That's how it's going on. In at t is equal to 0, when the universe ceased to exist, there used to be all the mass, all the energy was concentrated at this pinpoint, which suddenly expanded. Suddenly it started expanding, and it is still expanding. So that sudden expansion of those matter and energy is what is called Big Bang. Not much to understand in it. There was a pinpoint with all the energy and matter. All the energy and matter was here. Suddenly, at t is equal to 0, started expanding. This is called Big Bang. Okay. And t is equal to present. We have our universe, which is still expanding, but at a much lower rate than what it was when the Big Bang happened. That is the story of the universe, how it started. Big Bang is one of the theories which explains the origin of our universe. Do not go and answer that Big Bang explains the origin of life. It does not explain the origin of life. In a way it does, because when life will originate only when there is a universe. But what it precisely wants to explain is the origin of universe rather than the origin of life. So don't make this mistake of assuming that Big Bang explains the origin of life. No, it doesn't. It explains the origin of universe. Explain the basis of classification is what the next question is asking. Question number 6. So it's a very open-ended question. Explain the basis of classification. I think a lot of you might be able to answer it on your own. Firstly, you have to be specific what kind of classification. I'm going to assume it is taxonomic classification of organisms. Chapter number 2, right? Yeah, I think chapter number 2. Whatever. I will assume it's asked. This question wants to ask the basis of taxonomical classification. What all do we see in order to classify organisms? Instead of mugging up the answer, please try to understand. I'm asking you a question. If I give you Lego, you all know what Lego are, right? Or building blocks, those small pieces which you use to build stuff. If I give you like a bunch of them, all in different colors, shape, size, will you be able to classify them? If you use your brain, you will be. So what criteria will you use? It depends on you, how you want to classify them. If you want to classify them according to color, you can classify them according to color. Different shades of green, you put them in one. Different shades of blue, you put them into the other group. Different shades of red, you put them into the other group. Different shades of yellow, put them in one group. Now what you do is, those shades of green, further classify them. Shade number 1, shade number 2, shade number 3, shade number 4. You have like different shades of green, give them names and classify those pieces according to them. Similarly, do it for blue, do it for green, do it for yellow, do it for red. Then find one more criteria to classify them further. Maybe it could be the shape, it could be the size, it could be the weight, it could be anything. But you need a criteria for it to keep going. You can keep classifying it as much as you want. Now, in taxonomy, what we use is, we use a similar characteristics. Now we use similar characteristic. If you have like 2 organisms and you want to classify them, organism 1, organism 2, organism 3, organism 4. Now organism 1, organism 2, organism 3, organism 4, we have like, let's give them similar characteristic. Organism 1 has, I'll give it a 1 star. Organism 2 has like 3 stars. Organism 3 have 2 stars. Organism 4 has 3 stars. 3.5 stars. Let's assume 3.5. Now, organism 2 and 4 are really closely related. Organism 1 is really far to 2 and 3. Far away. Organism 2 is again as close to organism 1 as it is to 3 and 2, kind of. So if we classify them, if we make a picture mental tree, we would put organism 1 over here, organism 2, then organism 3 and 4, right? Organism 3 and 4 could be a related species, 2 related species. Organism 2 could be their genera. Organism 1 could be the phylum, right? But if you assume all of them to be different species, then how will you classify? Then we will give them a common ancestor. Common ancestor, kept dividing, you get organism 1 over here. Kept dividing somewhere in the middle. Okay, so 2 was somewhere in the middle between 3 and 4, right? So we will draw a simple branch for 2. See, I'll show you. We will draw a middle situation for 2. This is organism 2. This is organism 3 and organism 4. So organism 3 and 4 are like closely related. They have like a common ancestor before organism 2, organism 1 and all. Organism 2 is a bit more related to 1, a bit more related to 2, 3 and 4. Organism 1 is very far away from 3 and 4, but closer to 2. That is how you classify. You classify them on the basis of their common characteristics. Question number 7. How many length of DNA after DNA packaging? Okay, so be a question. How many length of DNA after DNA packaging? I really don't understand the question a lot, but I'll try to answer it. Question number 7 says, I think you mean to ask if this is a DNA and after I package it, how, okay, I'll just tell you about packaging. I think that should answer your question, whatever you were trying to answer. I'll tell you some details about packaging. This is a histone octamer. Okay. It's a histone octamer. It has like 8 balls with it. What you do is you pack your DNA on it. Now, how much turns and all? 2.5 turns. 2.5 turns, do you understand what it is? I will make it a bit more clearer to you. Suppose this is the histone octamer. Imagine it to be this. Now, what does 2.5 turns make or give you? You start from here. This is 1 turn. This is 2 turns. And this is 2.5 turns. You exit. That's how your DNA is packed. So if your DNA is in a length and you need to pack it, so this is how you pack it at the lowest level possible on a histone octamer. Now, a lot of histone octamers are themselves packed. Okay. Histone octamers are themselves packed into beads and into clusters. Those clusters are themselves packed into, chains. Those are packed as an arm of a chromosome, right? Then they are packed as a chromosome. That's how the DNA packaging works. The entire human DNA length of around 2 meters or something, 2.2 meters, if I remember it correctly, is packed into a small space, very small space in the nucleus of a cell in 23 pairs of chromosomes. Got it? The entire 2.2 meter length of DNA is packed into 23 pairs of chromosomes, which fit in a nucleus of a single cell. That's how compact this packing is. I hope that answers your questions. Question number 8 says, why is vegetative propagation practiced for growing some types of plants? All the plants cannot sexually reproduce. That's the simplest answer which I can give you. That is why vegetative propagation is used. Otherwise, you can think of some examples and some advantages of vegetative propagation. Firstly, it requires just one parent. You need one plant. You will be able to grow a lot of plants from this one plant. You don't need a partner to partner up and sexually reproduce. One plant is enough. What else? You will get the exact same characteristics from the parent plant in your offspring plant, because there is no sexual reproduction, there is no involvement of fusion of gametes, recombination, genetic variability is not there. You get an exact replica of the parent plant in the offspring. That is desirable to, nursery owners and all. Because you have a good parent plant, you can keep growing offsprings by vegetative propagation and you can keep selling those offsprings. Unlimited offsprings, unlimited possibilities to sell them. It is really convenient. Apart from that, what else? What more can you think for vegetative propagation? It is fast, right? You don't need actually to wait for the perfect time, because plants are not like animals. They cannot go and copulate. They have to wait for the wind to be perfect, for the season to be perfect, for the pollen grain to come and land on the stigma or any other, anthrozoids, oogonia and all that. They need to wait for the perfect time, for the perfect moment, for the perfect situation, for all of that to happen. That is eliminated in your asexual reproduction. You can reproduce, you can produce offsprings on your own. You don't need environmental help or anything. That could be one of the advantages. All right. Okay. Going on to the next question. What is the importance of molecular basis of inheritance? We just discussed so as we were just discussing, whatever disadvantages are there for sexual reproduction will be covered by molecular basis of inheritance. So now if inheritance has a molecular basis, what does that mean? What purpose does that serve? Firstly, your genes will be recombined. There will be crossing over. That will lead to genetic variability and creation of new species over time, which are better than the previous ones. That's why they are going to survive. That is Darwin's rule. So your species diversity will increase. Your survival chances will increase because variations will come and go. You will be able to create better species over time. Molecular basis of inheritance can tell us how fast that will happen, how slow it will happen, which direction it is going. It is disruptive, stable, or convergent, diversive. All that information is given by your molecular basis of inheritance. I don't think there is a lot of points on importance of molecular basis of inheritance. So I hope that these points are enough, should be enough for you to be able to understand. Question number 9, then question number 10. Explain human reproduction. Oh my God. It's a long process, okay? Human reproduction cannot be explained in 1 or 2 minutes. Takes a whole class, maybe not just one class, couple of classes for a professor, for a teacher to be able to explain human reproduction. I will just give you the jinx of it, how the reproduction happens. We have a male and a female. The female will contribute to the egg, ova. It is called an ova. It has 23 chromosomes. The male will contribute a sperm. It has 23 chromosomes. If I elaborate, 22 plus X, and it can have 22 plus X or Y, whatever. Now, the sperm and ova are discharged. The sperms are discharged during copulation, and the next target for the sperm is reaching the ova. Once a sperm reaches the ova, it penetrates the ova. The nuclei, they fuse and form something called a zygote. Okay, this zygote will start dividing. It will start dividing, and it will become 8 cell eventually, and 16 cell. It will become into something called morula. Morula will become into a cavity filled blastocyst. This blastocyst will go on, uterus, right? It will go on and implant itself in the wall of uterus, where it will grow further. Blastocyst gets implanted. It starts developing. It becomes trophoblast. Trophoblast will start organification, and it will convert gradually over months into a fetus. Fetus will get converted into a baby, newborn baby. This baby grows and becomes an adult. That's it. That's the jinx of human reproduction. And all these steps have like a lot of intricate details, a lot of details which you will go and learn even in your classes. Plus, as you ascend up in your higher studies, you are going to learn even more further details about each of this process, how the sperms are made, how the ova are made, how they come about together, how the zygote is formed, what happens in the zygote, how the fertilization occurs, morula, blastocyst, trophoblast, how they are different from each other, what similarities they have, what are their characteristics, how things can affect all of these processes, what are the features of a fetus, at which months, what all do you get to see, delivery, how it happens, and all of that you will learn in your higher classes. We'll move on to question number 11. What is radial symmetry and bilateral symmetry? It's a very basic question from animal kingdom. We'll try to solve it fast. Question number 11. Okay. What is a... Just a minute, guys. Sorry for the delay. We'll continue from here. Question number 11 says the different symmetries, radial symmetries and bilateral symmetries. Imagine an organism which you can you can divide this organism into two equal looking parts only through one plane, through its body. This is the organism. Only through this. Achha, this is not okay. Place something like right. Now this organism you can divide into two equal parts only through this one plane, through its body. Otherwise, you see, like this plane, it won't be divided. It won't be divided from here. It won't be divided from here. Only one plane exists, which can divide it into two equal looking parts. This is what is called bilateral symmetry. Now, what a radial symmetry is, imagine an organism. You can divide it into two equal parts by any plane, more than one plane passing through the body. More than one planes exist. This is what is called radial symmetry. Got it? Next, question number 12 says, what is cell wall? Okay, cell wall. If this is a plant cell or a bacterial cell, to describe a cell wall, this is a cell. The outermost layer of a cell is called cell membrane. Now, to protect this cell, you have an additional layer outside the cell membrane, which is called this whole thing, which is, protecting and creating the outermost layer of the cell in plants and bacteria is called cell wall. Aptly named cell wall, the wall of the cell. It stops like lot of materials from entering and exiting the cell. It has to be flexible. Actually, a cell wall is a characteristic feature of a kingdom. So for plants, the cell wall is made up of cellulose. For fungi, it is mainly made up of chitin. For Monera, okay, Monera, you can have any cell wall made up of a lot of stuff. Hemicellulose, cellulose, samsonites, and all that. Lipopolysaccharides, and all those peptidoglycans. So, okay, that was question number 12. What is central dogma of biology? So, you know about Watson and Crick, right? Question number 13 asks central dogma. It was postulated by these scientists called Watson and Crick. The Watson and Crick also elucidated the structure of the DNA. Now, what a central dogma is, it says key flow of information in a biological cell is it follows a specific direction. What is the direction? You have the DNA, which is usually the storehouse of all the genetic information because it is quite stable and it has a good storage capability. DNA will get what? Converted to RNA? No, that process is called something. It is called transcription. DNA will get transcripted into RNA. RNA will get translated into protein. Now, protein will go and carry out the function of the gene. One more thing is the RNA can be reverse transcripted into DNA. This whole thing, this whole schema is known as the central dogma of biology. This is how information is passed on in biological systems. This is how your genes work. This is how your body creates proteins. All of these are explained by this central dogma. Next question is, what are diseases? Now, it is a very simple definition, disease ka definition, if you ask. Any condition which alters the state of your body, it affects you physically, mentally, or emotionally, it's called a disease. It affects the normal functioning of your body, it is a disease. Normal physical, normal mental, normal emotional functioning, normal social functioning. So, a condition, what does the condition do? It affects what? Normal functioning. Normal, what kind of functioning? Physical, mental, social functioning of body is a disease. Now then, there are lots of kinds of diseases. You have transmissible, non-transmissible, communicable, non-communicable. You have bacterial diseases, viral diseases, pathogenic diseases, fungal diseases, parasitic diseases. Then you have morbid, non-morbid, chronic, acute, lots of different kinds of diseases. Moving on to question number 15, guys. What is a plant is trimerous or pentamerous? I think it says, what if a plant is trimerous or pentamerous? What does trimerous and pentamerous means? Means that the flower and its appendages are arranged in a multiple of 3 or 5. So if you have like 3 petals in a flower, in a plant, next you have like 5 petals in another plant. Both of them are different. This is usually a dicot. This is usually seen in monocots. Not just flowers. You will have 3 sepals. You will have 5 sepals. You will have like usually 3 gynoeciums, 5 gynoeciums, usually 5 androeciums, 3 androeciums, all that. This is how trimerous and pentamerous settings work. How is the study of plant anatomy useful to us? Okay, trick question. It is useful. All these biological studies are useful to us in a lot of ways. Plant anatomy, particularly, it helps us to, discern the internal structure of plant. We can see what's happening inside of plant, where all the things are going on and how are they working together to, sustain the life of this plant. That can help us, obtain our benefit. We can manipulate the plant, we can manipulate its anatomy, we can take advantage of its anatomy to manipulate its physiology. From its physiology, we can manipulate its metabolism and get different stuff that we need for our own benefit. That's how you exploit the anatomy of plant. I don't think there's much to explain in this. I think that is pretty much it. We'll move on to the next question, guys. Question number 16, done. 17. So 17 asks, name the hormone which is involved in pregnancy and lactation. So for pregnancy, remember this hormone called progesterone. So in pregnancy, it is secreted by placenta. Progesterone, P for progesterone, P for pregnancy. Progesterone will maintain your pregnancy state. For lactation, you have this hormone called prolactin. This prolactin, L lactation, L prolactin. This hormone will go in favor on your lactation. It is secreted by pituitary, anterior pituitary. Understood? Moving on to the next question. What is protista? And explain. Protista. So now in the 5-kingdom classification given by Whittaker, protista is the kingdom which has unicellular, eukaryotic organisms. These two criteria. Unicellular, the organism should be a unicellular organism. Multicellular are not included in protista. Plus, the organism should be eukaryotic. Prokaryotic organisms are included in a separate kingdom called Monera. Prokaryotic unicellular and multicellular both. But if there is a eukaryotic unicellular organism, it is included in protista. Next, question number 19. How many pollen mother cells should undergo meiotic divisions to produce 64 pollen grain? One pollen mother cell will produce 4 pollen grains. One will produce 4. So you need to produce 64. So of course, you'll have 16 pollen mother cells will undergo meiotic divisions to produce 64 pollen grains. That's your answer. Answer is 16. Now there's a catch here. To form a pollen grain, one pollen mother cell will form 4 pollen grain, but one oocyte mother cell will form just one oocyte. So for female reproductive parts, it's 1 for 1, but for male, it's 1 for 4. That's what you need to remember. So to produce 64 oocytes, you need 64 oocyte mother cells, right? Next, 20. Introduce digestive system. Introduce digestive system. I'll assume this is the digestive system of a human. So it's pretty easy. It starts with the mouth. You have the salivary glands in the mouth. From mouth, it'll go on to the esophagus, which takes it to the stomach. The stomach will again open up into a duodenum. The duodenum will open into small intestine, which will give way to large intestine, rectum, and anus. That is your digestive system for short. Now you have like different parts. Different parts have different functions. The mouth has a function. It has teeth, tongue. The main function of mouth is to masticate food, mix it with saliva, keep it going, keep food coming inside, sending it to the pharynx, to the esophagus, to go to the stomach. That's all for mouth. Esophagus only function is to deliver it to the stomach. Stomach may start digestion of proteins takes place in an acidic medium. It gets transferred to duodenum, where the medium changes to basic. Basic medium helps to digest the remaining carbohydrates, fats, and proteins, which moves to small intestine. Small intestine will absorb. It has three parts: duodenum, ileum, jejunum. Duodenum comes first, then comes jejunum, then comes ileum. Terminal part of ileum joins to large intestine. Which part of largest intestine? The part is called cecum. Cecum will give rise to ascending colon, which will go on to become transverse, and descending colons. Sigmoid colon will give rise to rectum. Rectum may the feces, whatever leftover part which could not be absorbed in the small and large intestine, it is stored in the rectum for you to pass it out through anus when you feel the urge. All right, so that was it for today, guys. The remaining questions and the new questions, I will answer it in a next session. So I hope you were able to understand. That was Ask Me Anything for biology. This is Anudh Mishra. Have a good day. Thank you.
Thanks, Anudh.
Thank you, sir.